So we're going to take questions later during the panel session, and now I'd like to introduce Dr. Jonathan Keats, who's the Director of Bioinformatics at TGen and the Scientific Director for Multiple Myeloma Research at the City of Hope.
Thank you, Leif, and I get the pleasure to talk to you about something that I may be the least qualified or the most qualified to talk to you about, and this is clinical trials. So as Leif mentioned, I'm the Director of Bioinformatics, which means I'm about as far away from touching a patient, except for when I'm in a room like this and somebody's not so far away from me. But I also give you a very unbiased opinion of clinical trials, and I think a lot of times today we've heard a lot about clinical trials during the course of the day. And people are probably asking some questions about it, and I'm not sure. We'll have a question here in a second, which maybe we can just bring up, and that'll probably start the conversation. We go to the next slide, or I guess I get to do that. So we have a quick question. Just clinical trial participation represents a generally more risky treatment. Is it only recommended for patients that have no other treatment options? Provides at least as good a treatment option as standard of care, opens access to promising new drugs. If you have a second to do that. I guess for those of you who don't know, generally in the US population, particularly in adults compared to children, the participation in clinical trials is fairly minimal, usually less than 20% of patients, which, especially if you think of how things have improved in myeloma over the last decade. So for me, I started in 1999 when melphalan prednisone was the standard of care. And today, that really wouldn't be something we would give to most patients. So we have the results for this, or do I have to go to the next slide? So the most common, it opens up access to promising new drug treatments that are otherwise not available. And I think that is true in many cases, though sometimes that's obviously bringing things into earlier lines of therapy also that we're doing with these patients. But I'm glad to see that people didn't select more risky, because that's, especially in the myeloma age today, I think people like myself and a lot of the other speakers who put a lot of effort into understanding this disease, the things that we bring forward to clinical trials today are generally not things that we have question if they work, and we spend more time fighting over which trial to do and which agent we think is the best, because there's more drugs to test than there are patients that are involved in trials. So the goal of a clinical trial is really to help us develop treatments and strategies to potentially lengthen lives. That's what we really want to do. We want to see if some of these new agents are actually working and try and develop these new medications to really get them from those patients who are at sites that are participating in clinical trials out to everybody in the country and the world wide. And why I would tell you to maybe consider participating in this is you've heard a lot today about how myeloma has changed dramatically in the last decade, 2 decades. And this is because of those patients who participate in clinical trials. Someone like Dr. Ken Anderson can test every one of these drugs in his research lab at Dana-Farber, but if a patient doesn't participate in the trial, we'll never get these to patients. So this is the way we get ultimately all this effort that we put into trying to identify the best way to help people into everybody out there in the community. We've had a lot of new drugs approved. We won't talk too much about that, so you've heard a lot. You probably got overloaded with that today. There are many new drugs being studied in clinical trials, and it does help us as we're starting to really tease in drugs that work for certain types of myeloma, and I'll talk a little bit about that more in a second. So what are some of the current research questions? And I think these are important as you're considering trials, and these are probably the things that we spend a lot of time fighting about, especially in the clinical meetings. One is, should we be treating people with smoldering multiple myeloma? We just had a discussion from a patient with smoldering multiple myeloma. We know these people have a chance of developing bone disease or developing anemia, at which point we would want to treat them. Maybe we should treat them in advance. If you're getting your colonoscopy, while they're doing your colonoscopy, if they see a polyp, they cut it out. That's the treatment. You get rid of the pre-malignant disease. We know you have MGUS, we know you have smoldering. We don't have the ability to cut it out, so we do have to consider, do we need to treat those people? And are there some people, could we treat them and prevent them from ever getting the disease and the complications that come with the disease? And obviously with things like the new immunotherapies, that is more of a possibility than ever before. We're also asking questions, what are the best treatments for newly diagnosed patients? We got a lot of new drugs, trying to figure out which order to put them in. If you talk to anyone about childhood leukemia, over the last 3 to 4 decades, you've seen people go from a overall survival rate of 5 to 10%, upwards of 90% now, really by just playing with the drugs that are available and figuring out the best way, the best combination, the best dosing, the best time intervals. Today we have this beauty in myeloma of having so many agents available, but there is going to be also a time when we're going to have to put a lot of effort into really figuring out how to combine them, the order, the timing, the dosing, to really get people to the highest cure rate possible. And then also, which treatments to match the patients and their genetic subtypes. There are certain treatments that we have coming today, like venetoclax, that we think really works better in certain myeloma patients than others. Now, if you're considering going into a clinical trial, obviously the best way to do this is to talk to your treating physician and the nursing staff at your clinical site. And if they do offer them, do take the time to read the informed consent. Some of them can be very painful and long, but it'll give you particularly an idea of the expectations we have from you in participation. Some of the clinical trials do have additional bone marrows required as part of the trial because it helps us understand what's going on in the trial. But it also just gives you a better chance to understand everything that's going to be asked of you. Now, what are some of the benefits? And I have 2 of these cute little guinea pigs that my daughter's made me get. So you will not be treated like a guinea pig. That's the most important thing to be taken care of. You will have normal standard of care treatment. We never give placebo in multiple myeloma. That sugar pill just does not exist. And you will have regular visits with your treating. You may even have additional care, investigational care with your patients. And I'll be honest, if you walked up to me and asked me, what should I do with my cancer? I don't care if it's any cancer. I will usually tell you to consider a clinical trial, largely because we do have to, there's requirements as a clinical trial. I won't say you'll get better quality of care, but there is so much mandated work that they have to check on you and monitor you. At the end of the day, you're being observed much more intently than the average patient. So there can be just some general benefit, and that's generally why I'd suggest a trial to people. You're being observed a little bit more readily. You will, and again, you will potentially from that have higher standard of care available to you. How do you figure out? Obviously, be careful with Google multiple myeloma clinical trials. But that's the simplest way. But obviously, ask your treating team. There are a lot of academic medical centers in the region that will have these that you can access. Clinicaltrials.gov with the National Cancer Institute is a good way to access what's going on, but particularly because that gives you every clinical trial and every indication. Some of the patient-led organizations, I'm not sure if AZMM has their own, but the Multiple Myeloma Research Foundation, the International Myeloma Research Foundation, these groups do really maintain lists of myeloma clinical trials that can help you identify which trials are available in the Southwest region, for instance, which ones are available in the US, so that if you're interested in participating, you can get access to this information. There are a lot of agents, and this just kind of goes to the idea that I was saying. We have to fight today to recommend which trials we're going to run, because we have way more agents to test than we have patients that can participate in trials. So we put a lot of effort in trying to just really nominate the best ones forward to give patients like yourselves the opportunity to participate if you choose to. Now, just as a quick summary, clinical trials really do drive research, and it's brought us to where we are. We wouldn't be talking about 10-year possible outcomes, threefold improvements in outcomes if people hadn't participated in clinical trials with things like the proteasome inhibitors and IMiDs. No one is treated like a guinea pig. No one gets a placebo. These are really taking, especially if you're in a phase 3 clinical trial in multiple myeloma, we are taking the best in class against what we think will be the best in class in almost every case, and they've been tested extensively already at that point in time. I was also asked to talk about something that's much closer to something I really deal with on a day-to-day basis, and this is precision medicine. And some of you have heard this term a little bit today. We've talked about it with the Multiple Myeloma Research Foundation COMPASS study, really building a foundation for precision medicine. So in just a couple of minutes, I'm going to take you a little bit through to explain to you when we talk about precision medicine, what are we talking about? And a lot of us use similar terminology, but you'll hear slightly different interpretations. But this is my simplistic view to present to you today. Most medicine, and definitely what we practice in multiple myeloma today, is what we would call evidence-based medicine. And this is basically saying, if we look at this picture today, there are the average group of patients, so this big group is all patients in the room. But you can decide if you're the red patient or you're the black patient down here, or you're happily maybe one of the patients up in this bucket. But today we give you the treatment, and this is the basis of evidence-based medicine, we give you the treatment that works for the most patients, because that makes sense. If I could tell you something's going to work for the majority of people in this room, most people are going to ask to have that. But we do know that that treatment is not necessarily going to work for everybody, but it works for the most people. And should that fail, we give you the treatment that we think works for the next biggest group of patients, and so forth and so forth. But if you're this patient with the black dot down there, you really would have liked to have skipped those first two treatments and gone right to the first one, give you the right treatment for your disease today. And if you're the patient in the red, unfortunately today we probably don't have an approved therapy that really is optimized for you, but maybe if you submit a sample, you get tested in many of the labs around the country, or you could lab like ours, we'd flag something unique about this patient and be able to say, you know what? We don't really expect you to respond to the standard of care treatments, but we have a drug that we're pretty certain is going to actually work for you, and we have clinical trials available for you with a drug tailored to something about you, personalized. And that's really the model that we're trying to move forward to. We've already in the myeloma field advanced into what we call risk-adapted therapy. So depending on certain features that we know about your disease, we try and give you therapy a little bit more tailored to your risk status of your disease, and that's been really pioneered by the group here at Mayo Clinic with their MSMART protocol. But as you've heard, multiple myeloma is not one disease. If you talk to somebody when you're at the hospital, you as the patient or you as a caregiver, and you meet someone with lymphoma, they always say they have a follicular lymphoma. I have Burkitt's lymphoma. Lymphoma is not one disease. It's a lot of different diseases. Multiple myeloma, today we still call it one thing, but depending on the category, all of us in the room would agree that as experts, there's probably at least 4 subtypes, maybe 7, maybe 12. But multiple myeloma can be broken into different groups. Now, I'll tell you the secret from a person like me who focuses on the genetics. The great news is the best-in-class therapies that we have today have been working for almost all the groups of patients. And that has been the savior today, that we have therapies that work for everybody. But as we start to get into the next stage, hopefully some of these new therapies will really work for different groups of patients, specifically as we break people out into different groups. But it is a heterogeneous disease, which makes you the question, is there a treatment that works better for certain people than others? And probably one of the best cases that's came out this way, and maybe will be one of the next drugs approved, is that there's a drug called venetoclax that's used to treat CLL. It works really effectively in this group of patients here, these 3 subtypes. And now we're trying to figure out, does it just work in these patients? And this is actually 3 different groups, or is it actually only working in this group of patients over here? And there's a lot of us in the field putting a lot of effort into understanding how do we identify the group of patients who are going to benefit from that drug most effectively so that we can get it to that group of patients earlier. So a little bit of questions for the audience and particularly the patients. A lot of this depends on profiling and characterizing your disease. So have you had any molecular characterization performed on your tumor as part of your clinical care, such as FISH, you might have heard that word, cytogenetics, I'm not quite old enough to remember when that was cutting edge, or sequencing or next generation sequencing, you may have heard of it, or have you had multiple of those tests performed as part of your clinical care? Okay, so 24% of you listed no. I'll be honest and say that scares me a little bit, but also the highlight of that is a lot of places wouldn't change your treatment based on the data. But it is nice to know that you have, it is nice to know you'd have it, but I'm surprised that many people don't have any information. A lot of people have had FISH, that's really the standard of care. 5, a lot of people have had multiple of them, and then the group don't know. And I think this is okay, because for the most part, we don't use it today to change your therapy. We will do some risk adaptation, so obviously in the centers that do that, they will ask to have these tests performed, and hopefully if you're changing centers, somebody will explain to you the value for their treatment strategies and having that information available. So next slide. So how do we do this process of matching people to the right treatments? And it was kind of mentioned earlier, but today the standard way we would do this is when you're having a bone marrow biopsy performed. We're going to take some of the tumor cells from the bone marrow biopsy, take it into the pathology lab or the advanced genetics labs, and try and understand those tumors by pulling out of the tumor cell the little X's or chromosomes. These are the DNA that you inherited from your mom and dad, and pulling it apart and then really actually sequencing or testing the DNA to understand how many copies of different genes do you have. Is there a mutation in your tumor? Is there a different type of genetic event in your tumor that lets us predict the therapies that we may try and get to you, or identify clinical trial arms that you may be most appropriate to? Now, something that's been really pioneered by the Multiple Myeloma Research Foundation is this COMPASS study. I run the scientific side of this here at TGen. If any of you are ever interested in coming and seeing what we do on multiple myeloma at TGen, please feel free to contact our foundation or our marketing communications team. They'll arrange for you to come in and get a tour. I'd be happy to meet with you and talk to you about what we do there. But this is really a landmark study. It's actually, in regards to cancer sequencing or DNA sequencing, it is actually the largest cancer genomics study in the world. You saw that in the video earlier. But to consider this is one of the more rare cancers on the planet, to think that we have probably one of the biggest wealths of data is an amazing feat. And what I'll tell you even better is because this is a disease where we can pull out the tumor cells from the normal cells, our data quality is, as I will argue with anybody, it's the best quality data of any tumor group out there. So we know more about this disease than I'd say almost any other cancer out there. So we're trying to really push the boundaries today in understanding this disease. The goal of this study is to really understand what different types of myeloma exist and how do those different types of myeloma respond to therapies. And we're just starting to get to the point where the patients have been treated long enough to really start to ask those questions effectively, and that's really coming out in this coming year. Now, one thing I just want to give you an example of this is two patients from the study, both females, early 70s, both had stage 2 disease, both treated the same, both had the 414 translocation, both had chromosome 13 deletion, both had 17p deletions, but one progressed in under 10 months, so in under a year. This patient still, as far last 2 months ago, we got an update, they're still 24 months out. No progression at this point in time. We don't know. It could go to 10 years for all we know right now. But what differs those two patients is actually this status of P53. The patient who progressed really quickly had mutated P53. This patient is wild type. And what I want to point out to you, and this is why I harp on this with all our clinical colleagues, is because this patient had 414, they're historically considered high risk. And particularly even in today's day and age, because they have 17p deletion or this event, they definitely got told they were a high-risk patient. So it's never nice when we're telling somebody you're high risk when you may not be. And what we know from COMPASS today is that it's these patients, we think this event is really centered on a gene called P53. We call it the guardian of the genome. It's kind of the master preventer of cancer and human cancers. And we're born with 2 copies of it. So we have good brakes in the front, we have brakes in the back in our car. If I hopped in a car with me and we're going to go whipping down the 101, if I was going down the 101 and I said, oh, by the way, the brakes only work in the front of my car, you might get a little freaked out, but you're pretty confident I might be able to stop and get off the side of the road when you say, wait a second, kid, get off the road. And that's a lot of those patients with 17p deletions. What we've seen in COMPASS today is now we can pull out those patients who have really lost both copies, or we've cut the brakes in the front and the back, which is a smaller 25% of those patients with the deletion. And it's that group of patients who are doing very poorly. So we have ways today to tell a patient that they're really high risk, and then to also prevent telling people that they're really high risk when they may not actually be. And these are important messages we need to get out into the community so people are getting the message correctly. Now, from those kind of approaches, we can also identify mutations that are in the tumors that tell us different therapies we may be able to give you that we think will work more uniquely in you or not. So we'll focus today on this one down here, BRAF. So what we found in a previous study that we've done in the Myeloma Genome Project, that about 8% of people have mutations in BRAF. This is a gene that's mutated in almost 100% of skin cancers, and they've made a drug for treating skin cancer that targets this mutation. So all those people that have skin cancer led to the development of a drug that we can now use in 8% of myeloma patients. And we're starting clinical trials now where we say, if you have this mutation, we're going to give you that drug that was developed and approved to treat skin cancer. No reason to use it because generally we wouldn't have known that these patients had that same mutation. So this is personalizing the therapy to those patients that have that mutation. And again, this is an example of one of the patients that did get that therapy. And if you're like me and you're a clinician, we're just going to pick one dot here to focus. You're not a clinician. Look up here in the right shoulder, really hot black area. At diagnosis, that's the tumor sitting there. After treatment with this targeted therapy to that mutation that we'd only give to 8% of the people in this room potentially, the tumor has now gone away from that region. So we've had a really effective therapy where we're personalizing and targeting it to the tumor itself. And that's really spawned on an initiative from the Multiple Myeloma Research Foundation called the MyDRUG Protocol. This clinical trial is very close to getting approved by the FDA and going into full effect, where patients will, and they progress, be sent from your clinical site, any MMRC site. They'll send your samples out to get sequenced and tested, and then patients will be eligible based on their data of their tumor to go into some of these different buckets to test drugs that are very specific to the defects or mutations that exist in your tumor, but maybe not in the patient sitting in the chemo chair beside you today. So just some summary of precision medicine. This is really with the idea that one-size-all treatment approaches for myeloma are inappropriate since we know there's a very heterogeneous disease, and it's more heterogeneous than almost every patient is unique. There's not two COMPASS patients who are absolutely identical. Everyone's got something that differentiates them. Efforts are underway to better understand the nature and the disease and to provide better personalized approaches. Genomic sequencing and data from myeloma patients are key to identifying the subtypes. The more we do, the more subtypes we really identify, and our attempt now is to try and make it, what are the clinically relevant subtypes? We can identify the number as high as we need, but it has to be clinically relevant. And participation in clinical trials is going to be the way we're going to get these drugs approved and tested. There's a lot of caveats to the way people like myself really drive these clinical trials to ensure they're done correctly. But we're really trying to push this, and especially in the myeloma space, I think we have the chance to push this very quickly. And thank you very much. I think we're going to move up to the panel probably now.
Thank you, Jonathan. I'd like to invite Dr. Larson and Dr. Anderson and Dr. Usmani to come to the panel. Take the questions with you, Ken. And I'd like you each to look at the questions and to choose a question that you think that you'd like to answer. And while you're going there, I'm going to ask Jonathan a question. Jonathan, does insurance cover the cost of clinical trials? Who pays for clinical trials?
So I'll try my best, and somebody can correct me if I'm wrong. So clinical trials, most of it, when it comes to the investigational drugs, you get that for free because the pharmaceutical company and the group funding the study is providing the drugs for free. So I'm a Canadian, if you can't get it from the accent. So we definitely would often encourage patients going to clinical trials because you could get drugs that just weren't available through the public healthcare system at all. So that was one encouragement. Obviously, sometimes when you're having certain tests done as part of the clinical trial, they may be covered by the clinical trial themselves if they're only being done for the clinical trial. But for ones that are part of standard of care that you would be getting your SPEP that you would get every month to test your tumor burden, that I think still gets billed to your to whatever coverage you would have because it's part of your standard clinical care. You'd have that done regardless if you're in the trial or not.
Has someone chosen a question? Ken, go ahead.
Yeah, so I've chosen that what is the M-spike number for achievement of MRD negativity? And there is no such number. It does stand to reason that if you have a large M protein that you probably aren't MRD negative because that monoclonal protein is being made by tumor cells somewhere. But in fact, many of the trials where we look for MRD negativity, there are patients that are included in those trials often who have a very good partial response, meaning a 90% decrease in their protein, but it's still there. and we look for MRD, and you say, well, why would you do that? And the answer is that, in fact, it's a new test. We need to compare it fairly, but we shouldn't make assumptions. But as an example, the half-life of the protein, it takes weeks for this monoclonal protein to go away. Even if the whole production were stopped today, it would take several weeks for it to go to half and half and half by each time. So it is conceivable that you could have MRD negativity in the bone marrow and still have a residual protein. So the point of the whole thing is that, honestly, MRD negativity will be an independent assay someday soon that will be clinically used, in my opinion, someday soon, but it's not going to be based on a monoclonal protein. It will be done in patients who have cleared the majority of their protein, probably, but it's not going to be exclusively in people who don't have a monoclonal protein.
Thanks, Ken. So now I'm going to give the speakers a warning. I'm going to go in order, starting in Jeremy. Jeremy, Barb, Saad. So Jeremy.
Sounds good. A lot of absolutely amazing questions. It's actually kind of overwhelming where to start. So I'll pluck one that I was just looking at. So the question is, in smoldering myeloma, there's a 10% risk of progression to active multiple myeloma. If you were not diagnosed with myeloma within the first 5 years, what are your chances to progression? So I think we lean heavily on some of the epidemiology data that's been put forth from Olmsted County in Minnesota by Dr. Kyle and the group there. And so what they had predicted was, after those first 5 years, that you actually morph towards more of that MGUS type of epidemiology risk, so closer to even 1%. Granted, I would still watch those patients very closely. There are a lot of factors. You know, when we talk about smoldering myeloma, really it's more than just looking at one number. It's trying to put the whole picture together. Is there a slowly evolving M protein? For someone that I see, and every time at 3 months they come in at 3.1, to me that's a very different story than every 3 months there's a 25% drift in that number. So there still are some variances, but I would say those first 5 years really are when we're watching most closely.
Barb.
Hi, I obviously can't answer any of the more technical questions, but I found a couple that were asked about how do people actually get in touch with their social worker or somebody who can help them really navigate the system. And yes, most of the larger institutions or cancer centers do have social workers on staff. And what I've found is that they are generally extremely helpful, but a lot of times patients aren't being referred to social work, and even though the PA or the other medical people often will want them to get additional help, there sometimes is a disconnect. So I hope there's going to be an effort of, the medical side recommending, because I think sometimes people are afraid to ask. There are also oncology social workers in the community, possibly, frankly, not enough. One of the things we've tried to do is identify resources, and you can call us and we will, help you find the right person in your area, because that's important too. I also noticed another question which I can't answer from a scientific standpoint, but I do have some anecdotal experience, and that is we worked for 7 years on the Navajo Nation, and what we learned there was that there was quite a high incidence of multiple myeloma that was considered a possible cause of, caused by environmental effects. There's the uranium project. A great deal of money was put into a fund to help people who had developed cancer, myeloma, but other cancers, and we found that the information wasn't getting out to the patients and families, which we did try to expedite. The second area was that a number of people were being identified as myeloma patients because of the high incidence of diabetes among the Native Americans, but on the Navajo Nation. And they had a project for diabetes, and that's where we saw, the actual diagnosis of myeloma coming from that. Mayo and a couple of other of the cancer centers who came up to the reservation with us were very helpful, but obviously there needs to be a more systematic way of connecting people to the resources. So that's been my experience with that.
Thank you. Saad?
All right. So what I'm going to do is, pick up a couple of question or themes and kind of answer a question. I think we've had a few questions about kind of stemming from what Jonathan brought up, the bone marrow biopsy. There were a few questions. If I'm having a bone marrow biopsy, how do I know that I'm getting genetic testing on the bone marrow biopsy? The way that you would know is to ask your physicians if they're doing it, and if not, request them to do it. The second question is COMPASS study. Is it still collecting data on new patients? It's been a few years since the COMPASS study actually closed to accrual, so we're not doing that, but there is a molecular profiling study that we are doing right now and actively accruing patients to it. So that study is available. And there's a theme around who covers the cost for clinical trials, and is it the insurance or do the patients have to pay out of pocket? For the most part, clinical trials are funded by the sponsor, the study sponsor, or the drug companies. All the experimental costs are covered by the sponsor of the study. So, there's usually no out-of-pocket expense for patients.
Ken?
So I have a couple of questions. I think I have a microphone still. So I'll just couple two of them. One, it says, why not give relapsed medications right away when you're diagnosed with myeloma? And then the one I think we could put with it is, how much does quadruple therapy cost? Is it covered by insurance and or Medicare? So the first question is really a good question. And what you've heard in multiple talks today is new things come along and they get tested in relapse refractory myeloma. I call it the 9th inning with 2 outs and 2 strikes, okay? And then they move to the 7th inning in clinical trials, and then they move to new diagnosis. So Revlimid and Velcade were started in advanced myeloma way back 15 years ago. Now, as you heard nicely from Leif, RVD, Revlimid Velcade Dex, is used very commonly as initial treatment. And he also mentioned to you that daratumumab is now having gone forward and gotten approved with Dr. Usmani and others in relapse myeloma, is now moving into being tested together with or without RVD, with or without daratumumab. Anyway, the point is, it's a lesson that we've learned in cancer many years ago in the solid tumors and leukemias, but we're learning it in myeloma. If you put together different kinds of drugs that have different mechanisms, and this question is very prescient, but if you do it early, then you can prevent, in many cases, the development of resistance. That was learned in antibiotics with tuberculosis. It's a principle from HIV, where there are at least 3 different activities put in one pill, and HIV has become a chronic illness. And so in myeloma, I think the question is very right. We are defining things when we have to in people who have really active disease that's really been refractory, but that's not the way it usually benefits most. It's adding it to a team very early on. And in lymphoma, we have what's called CHOP, which stands for chemotherapy, basically, and Rituxan, which is an antibody. And maybe in myeloma, what you heard today is we might have RVD agents, targeted treatment with an antibody, daratumumab or another one maybe as a standard. That's happening at the same time, and people are being genotyped who are getting those things as we're developing specific targeted genetically based profile treatment. Both need to go hand in hand so we can get to the time that Jonathan mentioned when we can actually deliver very specific therapy in the cases where it's mostly going to work. The second part I put together with it was the cost, and that's a really, really good question. And I don't have the answer, but I think we really have to be aware of this because Revlimid, Velcade, each of them very expensive. Daratumumab even more expensive. So it's not uncommon nowadays to have $30,000 or $40,000 US per month for therapy of combination targeted treatments. I'm not the one to decide what we should spend or not. But the point is, I think what I would love to see happen is we actually define medicines, actually they get reimbursed based on the outcome. And if that happens, if it really is a positive outcome, it gets richly rewarded, or whatever the right words are. If it doesn't work so well, it doesn't. And that's the first principle. The second one, I think, is if all the patients, not only in North America, but around the world, got the medicines that could really help them the most, everybody would win. We as caregivers up here would be much happier. I believe strongly the cost could come way down, and the pharmaceuticals would be happy because it would be much more broad use, and in fact, it would be a win-win for them too. So I think to me, the example that's going to force the issue is the CAR T cells. When we get into hundreds of thousands of dollars, and I think it's going to really push and make us address this in a more serious way. But I think together, you can count on those of us who are caregivers to be your advocates, because we want to do what's right for your health regardless of what the cost is. But in order to make that happen, we have to be responsible and make sure that access is allowed. The NCCN is one good example of that, where we have evidence-based things that get reimbursed if they have a lot of evidence and might not if they don't. But we need to get really very serious about this if we're going to start to use some of these new medications more routinely.
Thanks. Jonathan?
Well, there's a series of questions kind of relating to risk of family and how I got this and why I got this. So I think I'll kind of go through some of those. One, myeloma is relatively rare, and particularly it involves generally elderly people, although I'm too young to, maybe Ken will disagree with me. But for the most part, myeloma, though there are some clusters of myeloma in some families, largely we do not consider it to have much familial linkage. So the chance that your blood relative is going to get this disease is pretty low. The caveat being you have about a 2 times higher risk of getting myeloma as a blood relative of somebody else who has it, or other related blood cancers. But that means you're going from 4 in 100,000 to 8 in 100,000 for the majority of people in this room. So it's a pretty, you'd like to buy, that's a lottery ticket you probably won't buy because the chance of winning is pretty low. There was a question about incidence in Native Americans. It's not super well established, but generally we have a pretty good feeling that people of African ancestry have the highest risk. Caucasians, about half of that risk. Asians, about half of what the Caucasian population does in risk. So we kind of go from 8 in 100,000 for people of African descent to whites being 4 in 100,000, Asian descent 2 in 100,000, and Native Americans are most likely closer to the 2 in 100,000 range based on that association with ancestry just historically. Are there exposures that cause multiple myeloma? And the answer is, quote unquote, yes. We know if people who are exposed to Agent Orange, people who had a lot of radiation exposure are more likely to have multiple myeloma. They're also more likely to have a lot of other cancers. There's a bit of association to people that have been on farms and had exposure to pesticides. Again, that's probably the closer one to myeloma. But the general answer is, like, can we see, there was a question about how do we know what caused my disease? And for the most part, what we know is most of these diseases came down, there was one day in your body, there was a single B cell, and a tragic genetic error occurred in that single cell, and it's largely because those cells are doing amazingly beautiful things every day to provide you with your immune system, rearranging their genes to really provide you that function. And 1 out of every couple of 100 billion, they will mess up, and you had that one tragic error. So that's really, there's nothing you did. It was just that we have a very amazing immune system that provides us with a lot of our life. But every once in a while, a genetic error that provides that feature went wrong, and that's really what caused the disease. It wasn't anything you did. It wasn't anything you ate. It probably wasn't an exposure you had. For the most part, this really does seem like it's a random genetic event.
Okay. Thank you, Jonathan. I'm going to answer one. And it is, why is relapsed myeloma coupled with refractory? Why do we say relapsed refractory? And that's an important distinction. And it's probably, we get a little bit lazy and we don't distinguish them maybe as well as we should. But when you start, you've got newly diagnosed multiple myeloma, and we treat it. And then when the disease comes back, that's relapsed disease, which is better than when the disease is refractory, which means that it either doesn't respond to the treatment we give you, or if it does respond, it only responds for a very short time and comes back. And so of the three, the refractory is the worst. And when we enter patients on clinical trials, often we introduce drugs into the setting where none of the available drugs that we have work. And those are the patients with relapsed and refractory disease. Jeremy.
So I'll stick with the theme here. So I'm picking up a couple of questions pertaining to clinical trials and novel therapy. So one question mentions, how do our local oncologists become aware of new treatments? And so I think this kind of goes back to a little bit of what we talked about at the start, that really being an advocate for yourself is part of this. And so you're all here, so I'm preaching to the choir in some ways that you are kind of the cream of the crops. You're already doing that. But, honestly, I think this is making sure you're comfortable with your provider and that there is a dialogue there and you feel like you can ask these types of questions. And, speaking from my own personal experience, and I know the rest of the panel would probably agree that many times we're functioning in a consultative fashion where maybe you have a foot in with your local clinic and they're helping to be the boots on the ground. But when it comes time to, we're going to be on a new path and you're at that fork in the road, that maybe it is helpful to have that extra layer of expertise. And so that we can help, provide that extra piece of information as well. So I think there's multiple ways to go about this. And honestly, I think the community practitioners do an amazing job, and it's a very challenging thing to keep up with in myeloma. I feel like even when I'm looking, there's new trials that I wasn't aware of. So it's changing at a lightning fast rate, which is wonderful.
I'm just going to comment from the patient caregiver standpoint. The whole issue of clinical trials, which I learned a lot about that in the beginning from Mayo and from TGen, was that a lot of people didn't know what they were. They really didn't understand them. And I'm so glad because a lot of you have kind of repeated today, no, you're not going to be a guinea pig. Well, that was a feeling for a long time and a perception. But the other thing, it's very difficult as a patient sitting across from your doctor with whom you are very dependent and with whom you have a relationship to sometimes bring up the comment or say, by the way, I went to a conference and they were talking about clinical trials. Very often the community physician or oncologist is not up to date, which is understandable, but the problem then becomes that gap of awareness. And again, I unfortunately have seen the number of different practices often seem to be in a more compete rather than collaborate mode. So it's very difficult. So if you feel that you can, as was offered here by, TGen and I'm sure Mayo and other people are here, if you want to be able to talk with your own doctor about it, you certainly can ask for help from one of the resources. We really try and encourage you to ask for what you need. And I hope when you're here and get so much good information that you feel more empowered, that you can advocate. But I do appreciate how difficult that can be. So I just wanted to comment on that. The other area is we were talking about, and I was very glad that Ken spoke about, to me, the real elephant in the room. I get an average of 1 to 2 phone calls a week from people, from myeloma patients all over the country saying, I can't afford my medications. It's called prescription assistance programs, and also from the pharmacies aren't clearly either known by the patient or as accessible as they need to be. And what you're describing is medications, drugs that are going to become increasingly more prohibitive. And I really feel that we have to get together as ethical healthcare professionals and patients and say we have to advocate for collaboration or a way of helping people be able to continue the treatment they need to live. So thank you for bringing that up, and thank this whole group here of the most dedicated, wonderful people. I say this, I know, for all of you. So.
So I'm going to take this question that just popped up. Do oncologists sometimes work on a patient as a team? My answer is absolutely yes. That's how it should work. You guys are at the center of attention. We are just the supporting crew. Our job is to provide you with the best answers and help you manage your disease. And, there are patients who are seeing several of us, several myeloma experts at the same time, and we are consulting with each other in trying to get the best answer for that particular patient. So that's how it should work. It's actually highly encouraged.
Yeah. I agree with that. I always say when it comes to myeloma, the Red Sox and Yankees are on the same team. We're working together. In any event, sorry about that, but I picked two quick ones, and I'll be very quick. So one is, how often should I get a PET CT scan? And the other one is, what is bone turnover? And you might say, how are those related? Well, firstly, the PET CT scan is very useful. You heard today that in early stage myeloma, even people who don't have calcium in kidneys and anemia and bone problems on plain x-rays, even when none of those are true, if in fact they have a positive evidence with a sensitive test like a PET CT scan of more than one site of bone disease, they now get pulled out of a smoldering category into a group of patients that we treat. So it's really important there. And secondly, in our view, it is a very sensitive technique, not that specific, but we use it perhaps sometimes annually in patients just to be sure that in fact there isn't some occult problem. In those of you who are doing very well, and there are many, many, which is a great thing, sometimes myeloma can pop up in sites we don't expect, extramedullary, outside of the bone marrow. And really the only way to find that would be with a PET CT scan. And it's related to bone turnover because there can be low amounts of bone turnover, bone resorption, even when we don't see anything on the x-rays that we might see on the PET CT. And in the bone turnover question, real fast, Zometa, Iridia, early 1990s, used in everybody, as you heard, for 2 years so nicely, to stop or decrease bone turnover. It kills the bone resorbing cells called the osteoclasts, these little Pac-Men that resorb bone. The thing that we should mention quickly is there's a new drug on the block, a new kid on the block. Rank ligand is a fertilizer for osteoclasts, so they love this rank ligand and they resorb more bone. But there's an inhibitor called Xgeva that has just been FDA approved to decrease bone turnover in myeloma. And the good news there is it's safe to give in people whose kidney function isn't normal. So that's a big deal in myeloma. But bone turnover is simply too much bone breakdown, not enough new bone formation. And we had Zometa, now we have Xgeva to help patients try to more balance that process.
I'd like to thank everyone on the panel for all their contributions.